Today, Jake and I are going to be telling you about new results from a new study, the Australian Autism Biobank study. And the way we're going to run this is that I'm going to give a brief introduction and then really pass over to Jake to give most of the talk. So we're talking about a study in Australia, and I know most of you know where Australia is, but here's a world map. You probably don't usually see the world map in this way, but I guess it's the way we see it, with Australia in the middle. And really just to emphasize that, the landmass of Australia is actually very similar to the landmass of the US, but our population is, 13 times smaller. And so why am I telling you this? I think it's really because the way in which we approach science in a smaller country like this means that we have to, obviously our research funding is also scaled proportionally. So we have to kind of think carefully with the research funds that we have in the way in which we can contribute to international research. So the project we're talking about today was kind of brought about by the Autism Cooperative Research Centre. So in Australia, this is a government partnership scheme, the Cooperative Research Centre scheme, which tries to bring together researchers with end users in a sort of translational type of research. So the Autism CRC has its vision is to see autistic people empowered to use their diverse strengths and interests. Their mission is to motivate, facilitate and translate collaborative autism research across the lifespan and the spectrum. I'm telling you this because I thought you might be interested in the way in which the CRC is set up. You can Google search them and find the website and see all the interesting things that they're doing. And they have three key programs: the early years program, school years program, and adulthood program. So back in 2015, as part of their early years program, they told us that one of their goals was to develop a diagnostic protocol integrating behavioral and biological information to improve early and to improve accurate prediction of developing autism spectrum disorders. And so they came to us and asked us to help them towards that goal, telling us that they thought that within Australia, within the time frame available, that we had the capability of collecting data on about 600 families. So our immediate response was to say, well, this is a really important goal and one that many people around the world are working towards, but it's also a very difficult goal. And we had to say up front that we didn't think we'd be able to help them really achieve that goal fully in the time frame, but we would help design a study which would help along the way and contribute to international research. So, why did they come to me and my group? I guess it's because we have a reputation of research at that interface of statistics and genetics and applications to psychiatric disorders, and working particularly in the genetic space. So we work as a multi-group leader group. This is our group today, not back in 2015. And this is just showing some of the sort of work that we've done. So, for a long time, we've been working in that space of using genetic data for prediction of risk. With the first application of genetic data in psychiatric disorders. I work very closely with the Psychiatric Genomics Consortium. So this is an example of a study I'd led back in those days. This was the first study to estimate the genetic relationship between psychiatric disorders from directly measured DNA data. And we also, work very much in the theoretical modelling space as well. So this is an important paper from the group where we were looking at this important question from a methodological point of view. So when the Autism CRC came to us, the first thing I did was sat down with three key people. And I'm mentioning that now because these three people have kind of stuck with the project the whole way through. So the first was Gratten, who at that time was a senior postdoc in my group and now is his own lab head. Angeli Henders, who is an absolute expert in everything to do with data collection and project management and generation of any sort of omic data. And Chloe Yap, who at that point was a first-year undergraduate student who was very enthusiastic and passionate about genomics and disorders of the brain, and she had volunteered to come in our group. Chloe's now a third-year MD PhD student who has analysed the data from this project. So we didn't have a track record of working in autism spectrum disorders. And so the first thing we did was a literature review. And because we work in the genetic space, we reviewed the literature in the genetic space. So this is a summary, really, of 15 years of research, of psychiatric genetic research in one figure. I like showing this figure. It's a very nice summary. It was obviously published in 2016 after we were doing our own literature review, but the conclusions we drew were the same. So genetics research of the last 15 years has been powered by advances in technology, which have allowed different sorts of experimental paradigms, which have allowed discoveries of different sorts of genetic contributions to disorders. So on the x-axis, we have the proportion of variation attributable to different types of genetic factors. And on the y-axis, we've got psychiatric disorders of the brain, which are kind of ordered by age. So presentation in very early childhood through to presentation in adulthood, through to presentation in late adulthood. And the different colored bars represent the different types of genetic contributing factors. So we can use the technology of sequencing, and we've used this to identify new mutations which occur in a child and not found in the parent. And these studies have found that these sorts of variants really are predominantly found in disorders presenting in early childhood. And we've also used the genome-wide association study paradigm, which is a technology which is very cheap to generate genetic data and allows us to look at the DNA variants which commonly vary in the population, recognizing that every single one of us carries risk variants for every single common disease and disorder, but looking at the things which are more associated with people who carry a particular trait. And this graph very nicely shows that the adult disorders, the contribution from the common genetic variation predominates. Those early onset, early childhood presenting disorders really feature these de novo variants, and autism spectrum disorders is a mix of those sorts of genetic architectures. So when we were trying to make our decisions as how to conduct our study, and recognizing that we had a budget for that study, we had to work out, would we contribute to these studies in terms of the whole exome sequencing type approach, or would we try and use the money in a different way? Now, we realized that very large sample sizes were needed to generate these results. These are the sample sizes which are today rather than back in 2015, but showing this is the order of magnitude. Very large samples have been used to generate these results. And so with our 600 families, although we'd be able to contribute, in order to be leading new research, we thought we had to think of something more novel. So the other part of our literature research was to recognize that there was great heterogeneity and co-occurring conditions in autism spectrum disorders. And again, many people on the call are experts on this, whereas I am not. But it's clear that the complexity, we know that sometimes autism spectrum disorders are diagnosed together with intellectual disability. And yet we also know from genetic studies that the genetic contribution to autism spectrum disorders is actually very highly correlated with educational attainment and high IQ. So there's a great complexity and heterogeneity. There's many co-occurring conditions. And again, many of you are much more experts on this than myself. But our conclusion was that if we developed a study, that we would focus on deep clinical phenotyping and also a systems genomics approach. So to try and generate different types of genetic data. So not at first go down that whole genome sequencing route, but look at other sorts of genetic and genomic data that we could generate towards this goal of a diagnostic protocol which would integrate behavioral and biological information. And so really Jake Gratten is the person who has seen this project through from the beginning, through all the steps. And anyone who's been involved in data collection and project management will know all the things that go into that. It's been a massive effort and necessarily involves many, many people. I'm very pleased to pass over to Jake to describe our study and some of the results that are coming out of it. Thank you.
Okay, well, thank you so much, Naomi, for setting the scene. And thank you, Pam, and all of the team for this opportunity to present as part of the Simons Foundation lecture series. Good evening, everyone. It's early morning here, but I know it's late afternoon for you. It's really wonderful to be joining you. So I'm going to start my part of the lecture by telling you about the Australian Autism Biobank study in more detail, and then I will devote a majority of my time to describing a particular study that we performed in the biobank, which has generated some really interesting results, and that is on the relationship between autism and the gut microbiome. So the Australian Autism Biobank, as Naomi mentioned, is an initiative of the Autism Cooperative Research Centre. Recruitment began in the second half of 2015 and continued until the middle of 2018, by which time roughly 2,500 people had consented to take part. So this was a huge effort, and it was led by Andrew Whitehouse at the Telethon Kids Institute in Perth, along with Cheryl Dissanayake in Melbourne at the La Trobe University, Valsa Ethan at UNSW in Sydney, and both Helen Huesler and Paul Dawson, who are based here in Brisbane. So deep phenotype data was collected on participants along with a range of biospecimens. And all of those biospecimens were shipped to Naomi's lab in Brisbane at the University of Queensland, where they were receipted and processed by the UQ Human Studies Unit, which is led by Anjali Henders. We've subsequently generated a number of different genomic data sets in the biobank. And all of this data has been analysed by a very talented MD PhD student who Naomi introduced, and that is Chloe Yap. And I've highlighted Chloe here because I really want to acknowledge her up front. All the results that I'm going to present today on the gut microbiome have been generated by Chloe. Okay, so a little bit more about the phenotype data that was collected and the biospecimens that are available in the biobank. So of the roughly 2,500 participants in the cohort, nearly 900 are children on the spectrum, and there are just over 500 families in which data and samples are available for both parents and at least one child. In the children on the spectrum, data on autism symptom severity, social communication deficits, and restricted and repetitive behaviors was obtained using the ADOS. Adaptive behavior was measured using the Vineland, and sensory function was assessed using the short sensory profile. A number of different biospecimens were collected in these children, including a blood sample from which we obtained plasma, serum, buffy coat for DNA, and red blood cells. And then a subset of these children also provided a stool sample, a urine sample, and a hair sample. The same biospecimens were collected in children without a diagnosis, including siblings and unrelated controls. And phenotype data in these children included the social responsiveness scale, which is an autism-related measure, and information on developmental milestones. All of the children completed an age-appropriate IQ test. Data was collected on physical measurements. Parents completed a family questionnaire, and we collected information on medical histories and co-occurring conditions. Additionally, in those children that provided a stool sample, the Australian Eating Survey was completed to provide information on diet, and the Bristol stool chart was completed at the time of stool collection to provide a measure of stool consistency. The parents completed the Broad Autism Phenotype Questionnaire and the Communication Checklist, and parents of children on the spectrum also did an IQ test and also provided physical measurements. And a blood sample was collected from the parents. So that's a snapshot of the data and biospecimens that are in the biobank. It's a really valuable new resource that's available to the autism community, both in Australia and internationally, for investigating the biological basis of autism. So a few comments about the genomic data. As Naomi mentioned, we were asked by the Autism CRC to explore the utility of different types of genomic data as biomarkers for early and accurate diagnosis of autism. Acknowledging up front, of course, that the scale of the Australian Autism Biobank is not sufficient to address this very challenging objective in isolation. So the first data set that we generated was on genome-wide SNPs. This was we obtained genome-wide data on all participants in the biobank using the Illumina Global Screening Array. I won't go into detail about these data, except to say that we've described the data, including analyses of polygenic scores and copy number variants, in a paper that's been published in Molecular Autism. The second data set that we generated was on genome-wide DNA methylation. This was in blood-derived DNA. And here we used the Illumina EPIC array, and this was in just over 440 child participants. Our analyses of these data are still in progress, including meta-analyses of our AAB data with other data sets that are available internationally. But the preliminary results suggest that there's negligible association of DNA methylation with autism diagnosis. Another study which is still in progress involves generating metabolomics data in plasma samples. So we are currently screening a large panel of lipids and smaller targeted panels of neurotransmitters, acylcarnitines, and bile acids in plasma samples from a large number of child samples. And then finally, of course, we have performed a study of the gut microbiome, and this has involved shotgun sequencing of just under 250 stool-derived DNA samples from children for whom we have linked psychometric, genetic, and dietary data. And so this is the study that I'm going to focus on for the remainder of the talk. Before I do that, I'd just like to highlight that the data, including the phenotype data and the genomic data that I very briefly described, is available for autism-related research by application to the Australian Autism Biobank Data Access Committee. So this is the website here. If you are interested, I invite you to log in and please make use of this wonderful new resource. Okay, so I'm sure that most of you are familiar with the gut microbiome. For those of you who may not be, this is a term which refers to the very diverse community of bacteria and other microorganisms that inhabit the human gastrointestinal tract. There's been an explosion of interest in the microbiome in the past decade, as we have come to appreciate the important roles that this community plays in many aspects of human health and disease. Beyond the obvious roles in relation to digestion and metabolism, there is evidence to suggest that the microbiome plays a really important role in the development and maintenance of the immune system, and also that beneficial bacteria play a crucial role in preventing invasion of the GI tract by pathogenic bacteria. When the system breaks down, when commensal bacteria are disrupted, leading to dysbiosis, then this is often observed to co-occur with disease states such as inflammatory bowel disease. So why are we interested in the gut microbiome in autism specifically? There are a number of good reasons. The first one is that there's a high prevalence of gut conditions like constipation and diarrhea in autism compared to the general population. Another reason is that there are an increasing number of reports of direct associations between autism and specific bacterial taxa. A third reason is that there are also a growing number of reports from animal models of autism, some of which suggest a causal role of the gut microbiome on autism-related behaviors. And some of these studies, which are high profile, involve transplantation of fecal microbiota from human study participants with and without autism into germ-free mice. More recently, we've seen progression to human clinical trials involving this technique, so fecal microbiota transplantation, as an intervention for autism symptoms. So overall, there's really a great deal of interest in the microbiome in autism, both in the research field and also in the broader autism community. At the same time, there are some reasons to be cautious about how we interpret the available literature, and I've listed some of them here. So first of all, many of the studies that have been published to date are quite small in size, and so are likely to be underpowered. Another issue is that although a number of taxa have been reported to be associated with autism, relatively few of these robustly replicate across independent data sets. Thirdly, there are a number of important confounders of gut microbiome variation, such as age and diet, medications, for example. But there's variable correction for these confounders across studies. And this is also true of the need for multiple testing correction in analyses of differential abundance of bacterial species, for example. Another issue is that although some of the most high-profile reports of associations between the microbiome and autism have come from animal models, it should probably be acknowledged that rodents are imperfect models for human physiology and behavior, particularly for conditions as complex and heterogeneous as autism. So overall, there's a great deal of hype, I would say, in the field, but we question whether this is supported by the available evidence. And that is exemplified by a recent PubMed search that Chloe performed, in which she identified over 50 reviews on this subject, but the most recent only refers to 26 primary articles. So with all this in mind, and putting the hype to one side, a sober assessment of the literature leads us to believe that there are a few fundamental unresolved questions in the field. And the first of these is simply whether or not there is an association between autism and the gut microbiome at all, after accounting for confounding factors like diet. The second question is, if there is an association, then is this cause or consequence of autism? So these two questions are what motivated us at the outset to include collection of a stool sample and of time-matched dietary data into the protocols for the Australian Autism Biobank. And the objectives of the study that I'm about to describe to you were to attempt to address these two important questions, at least to the extent that was possible with the available data. Okay, so let me describe to you the study design. So we decided on a nested case sibling case control strategy involving just under 250 child samples. The case control sample included 99 children on the autism spectrum, all from the Australian Autism Biobank, and 97 children without a diagnosis who were unrelated, approximately half of whom were from the AAB, with the remainder coming from another Brisbane-based cohort called the Queensland Twin Adolescent Brain Study. So it's important to note here that the stool samples in the QTAB cohort were collected contemporaneously with the AAB recruitment effort, using exactly the same protocol preceded by Angelique's team at UQ, using the same protocols, and all of the samples were sequenced jointly into randomized manifests. So nested within the set of 99 autistic children were 51 for whom we had a stool sample in a sibling without a diagnosis of autism. So the case sibling comparison involves 51 autism sibling pairs, all from the AAB. So these 247 children were selected from over 500 with the goal of matching by age and sex. And you can see the distribution by age on the left-hand side here in each of the three groups. And here we can see the breakdown by sex. So there are no differences in age between each of these three groups, but there is a slight male bias, as is very common in autism studies because of the strong male bias in diagnosis. So all of these children completed the Australian Eating Survey to provide information on diet, and we prioritised children into the study with complete information on genome-wide SNPs and also psychometric data. All of these children are of European ancestry, and that is not because we are especially interested in Europeans. It simply reflects that that is the most prevalent ancestry in the biobank, and we wanted to limit sources of heterogeneity as much as was possible. Okay, now moving on to the data. So we partnered with a UQ spin-off company called Microba to perform whole metagenomic shotgun sequencing of stool-derived DNA samples. And this was performed on Illumina NovaSeq, and we generated a minimum of 3 billion base pairs of sequence data per sample. Subsequently, these sequence reads were downsampled or rarefied to 7 million reads per sample, and read space profiling was performed to a large database of complete bacterial genomes, so several thousand genomes. So this yielded two different data sets, a taxonomic data set comprising just over 1,700 bacterial species and several hundred viral species, and a gene level data set of over 5 million genes annotated to a number of different gene annotations and pathway annotations. We then performed some quality control, including removal of rare microbiome features defined as those with fewer than 10 non-zero values in the dataset. And this resulted in just over 600 species and approximately 2 million genes. All of this metagenomic data is linked to a large number of phenotypes in the biobank, including information on diet, available from the Australian Eating Survey, stool consistency via the Bristol stool chart, and a large number of behavioural phenotypes, including psychometric measures related to autism, sensory function, cognitive performance, and also polygenic scores for autism and related traits. And of course, we have data on age and sex. So just a few more comments on the dietary data, because diet is such a fundamentally important confounder of variation in the gut microbiome. The Australian Eating Survey is a validated food frequency questionnaire from the University of Newcastle here in Australia. The raw data comprises over 120 food level items, and from these data, there are a number of derived variables. One is the Australian Recommended Food Score, which is a measure of dietary quality, and another is percent energy measures for core and non-core food groups. So the core food groups are healthy foods like fruit and vegetables, and non-core food groups are unhealthy foods like confectionery and takeaways and so on. So when we perform principal components analyses on these percent energy food groups, the top 3 PCs explain over 40% of the variance. And as you can see, I hope on the left-hand side here, which is showing the loadings of the different food groups on each of these top 3 PCs, they are broadly interpretable in so much as PC1 is very high in healthy food groups like fruit and vegetables and low in junk foods. PC2 is high in dairy and low in grains and takeaways, and so on. And so I just share this with you because we very carefully adjusted for diet in all of our analyses using either the Australian Recommended Food Score or the PCs based on PCA of these percent energy food groups. Okay, moving on to the analyses proper. The first question that we asked was, how much phenotypic variance for liability to autism diagnosis and other traits is associated with the gut microbiome as a whole? So the rationale here was that this provides a putative upper bound on the predictive value of the microbiome, keeping in mind that one of our objectives was to explore the utility of the gut microbiome as a biomarker. The method that we used for these whole microbiome analyses was a mixed linear model approach using a tool called OSCAR that has been developed here in Brisbane in Naomi's group. So this involves the generation of omics data-based relationship matrices, or ORMs, based on different components of the microbiome data. In our case, we built ORMs based on the species level data and also the gene level data. And we also created ORMs based on common species and rare species, and also common genes and rare genes. And then because we were interested to know if autism diagnosis was associated with diet as a whole, we also built an ORM based on the dietary food level data from the Australian Eating Survey. Once an ORM has been constructed, it's possible to estimate the variance associated with that ORM in a mixed model framework using restricted maximum likelihood. And we refer to these estimates as the microbiome association index, or B-squared. So this is very much analogous to estimating heritability based on a genetic relatedness matrix that has been constructed using SNP array data in a tool like GCTA, for those of you who are familiar with those methods. An important distinction, of course, is whereas heritability represents causality, B-squared estimates representing the association of the microbiome with a focal trait like autism diagnosis may be cause or consequence of autism. Okay, so the first step that we took in this series of analyses before looking at autism diagnosis itself was to analyze a benchmarking trait. And in our case, that was age. We chose age because a recent large human gut microbiome study that has been posted on bioRxiv from Daphne Rothschild has reported, using methods similar to those that I've just described, that 28% of the variance in age is associated with the common species microbiome. So on this plot, which is showing on the x-axis the proportion of phenotypic variance for age, that estimate is shown by the dotted blue line, just here. So we wanted to confirm that we could recapitulate this estimate in our data set. And what you can see in this panel are estimates for the proportion of variance in age in the Australian Autism Biobank sample based on different ORMs, including for common species, rare species, common and rare genes, and for dietary variables. And happily, if you focus on the reddy pink estimate at the base of that figure, what we see when we look in the Australian Autism Biobank is that 33% of the variance in age is associated with common species microbiome. So that was very reassuring because it suggested to us that our data and methods are robust. And so on that basis, we proceeded to perform similar analyses of the liability for autism diagnosis. And that's what I'm showing here. And I think what you can immediately see is that irrespective of what component of the microbiome we used to make an ORM, there was negligible evidence for any association with autism diagnosis. So if we focus again on the common species microbiome results at the bottom, in sharp contrast to what we see for age, where over 30% of the variance is associated with the microbiome, for autism diagnosis, the estimate is 0%. Interestingly, the only ORM that was associated significantly with autism diagnosis was the one based on the food level data from the Australian Eating Survey. We also looked at a number of other traits that intuitively are more related or more closely related to the gut microbiome, like stool consistency and diet PCs. And here, although the estimates are variable depending on which component of the microbiome we look at, there are large and significant associations with the microbiome for these traits. So for example, if we look at stool consistency, 40% of the variance in this trait is associated with the rare species microbiome. And for diet PC1, 13% of the variance for this trait is associated with a common species microbiome. So the take-home messages from this series of analyses are that we see negligible association between the microbiome and a whole and autism diagnosis. And this contrasts sharply with what we see for other traits like age and stool consistency. The other take-home is that in contrast to those results, we do see a significant association between autism diagnosis and diet as a whole. And that's really crucial. And so I'd ask you to hold that in your minds because we'll come back to that later in the talk. Okay, moving on, the next question that we asked was there any evidence for differential abundance of individual species in autism? And for this, we use a method called ANCOM, which stands for Analysis of Composition of Microbiomes. This is a non-parametric method, which is acknowledged to be a robust approach to this type of analysis. We analyzed over 600 species and we adjusted for age and sex and dietary PCs. And because microbiome data is compositional, we used a centered log ratio transformation, which again is accepted as a robust approach to the challenge that is presented by compositional data. So moving on to the results, and just looking at the figure on the right-hand side, what this is showing on the x-axis is the center log ratio transformed mean difference in abundance between the autistic children and the combined sibling and unrelated control sample for each of 600 species. And on the y-axis, we have the ANCOM W statistic, which is a measure of the strength of association. The horizontal dashed lines show thresholds for significance, and typically values greater than 0.7 are used to declare experiment-wide significance, accounting for multiple testing. So what you can see overall is that there is relatively weak evidence for association of individual species with autism diagnosis, with a single exception, which is a species called Rambutia timonensis, which hasn't previously been reported to be associated with autism. So this species is less abundant in children on the spectrum compared to their siblings and unrelated controls. We also performed similar analyses at higher taxonomic levels, collapsing the species level counts to genera, orders, and phyla. And we did this because we were keen to determine if we could replicate published associations in autism. And what we found was that we could not. So there was no evidence for any association, for example, with the genus Prevotella, the genus Bifidobacterium, or the phylum Firmicutes, which is notable because our data set is by far the largest of its kind to date. Okay, so the next question we asked was whether there was any association between autism and diversity in the gut microbiome. And for this, we looked at two commonly used diversity measures, which are alpha diversity and beta diversity. Alpha diversity is a within individual metric, so it's looking at the number of species and relative abundance of those species within an individual community, whereas beta diversity is a between group diversity metric. So looking first at alpha diversity, we measured this using the Shannon index, which takes into account both the number of species and relative abundance. And we adjusted this for age and sex, working with center log ratio transform data again. And as you can see in this plot, we found no association between the adjusted Shannon index for alpha diversity and autism diagnosis. And that was also the case when we looked at beta diversity. Beta diversity was estimated using a weighted unifrac method. So this is taking into account the phylogenetic relationships between the taxa. And there was no association of pedodiversity with autism. So some take-home messages at this roughly midpoint of the talk are that when we look at the relationship between autism and the gut microbiome in the AAB cohort, we see negligible evidence for any association, I guess with the one exception of the association with Bombutia timorensis. And this is in sharp contrast to what we see for other traits like age and stool consistency, where there were large and significant associations with microbiome components. On the other hand, we do see a significant association between autism and diet as a whole. And so this observation kind of motivated Chloe to think about the data from a different angle. And specifically what she hypothesized was, if autism diagnosis is associated with restricted and repetitive behaviors, which are one of the diagnostic pillars of autism, then might this manifest in the form of restricted and repetitive diet, consistent with anecdotal reports that some children on the autism spectrum have fussy eating habits. And if that was the case, then might this indirectly influence the gut microbiome? So to investigate this hypothesis, the first question we asked was whether autism diagnosis was associated with dietary diversity. And to address this question, we calculated the Shannon index, so a measure of alpha diversity, on the food level data from the Australian Eating Survey, instead of making a Shannon index on the basis of species level data. And so I'm showing the distribution of these food level Shannon index estimates here for each of the three groups. And again, we have adjusted for age and sex. And what you can see, I think, quite clearly in the plot is that there is significantly reduced dietary diversity in children on the spectrum compared to their siblings and the unrelated controls, and this is highly significant. We also performed this analysis on the Australian recommended food score data, which is a measure of dietary quality, and we see very consistent results. So evidence for lower dietary quality in autism compared to siblings and controls. So that was strong evidence that dietary diversity is reduced in autism diagnosis, but we really wanted to stress test this result. And so the next question we asked was whether quantitative autistic traits, which were measured in the biobank, are also associated with dietary diversity in the expected direction. And so for this, we first looked at a number of different psychometric measures, including restricted and repetitive behaviors, which were available from the ADOS in all of the children on the spectrum, and also the SRS and the short sensory profile. So the SRS is available in both children on the spectrum and children without a diagnosis. And the rationale for analyzing the short sensory profile was that sensory sensitivity might also manifest in the form of restricted dietary preferences. And so what you can see, I hope, is that looking at the restricted and repetitive behaviors, is that there is a significant negative association between RRBs and dietary diversity. So more autistic-like behavior is associated with a more restricted diet. And that is also what we saw when we looked at the SRS and the short sensory profile, although in both of these cases, the association, although negative, was just suggested. Okay, so the next question we asked, trying to break this association or to make it go away, was to ask if polygenic scores as continuous genetic proxies of autism-related traits were also associated with dietary diversity in the AAB. And we looked at first a polygenic score for autism spectrum disorder, and that was using summary data from the largest psychiatric genomics consortium genome-wide association study for autism that was published in 2019 in Nature Genetics. And what you can see, again, consistent with the hypothesis, is that polygenic scores for autism are negatively associated with dietary diversity in our cohort. We then moved on to build a polygenic score for restricted and repetitive behaviors. And for this, we used summary data from a cross-trait GWAS for ADHD, ASD, Tourette's, and OCD. And before analyzing this polygenic score in relation to dietary diversity, we confirmed that it was associated with restricted and repetitive behaviours in the Australian Autism Biobank overall, in a sample of 868 children on the spectrum, and it was significantly associated. So it is a valid genetic proxy for RRBs. And what you can see again is that there is a negative association between dietary diversity and this polygenic score, although again it is suggestive. So just sort of summing up this part of the study, what we see is very consistent evidence or consistent signal that we can't make go away, that autism and autism-related behaviours are associated with reduced dietary diversity. And this is consistent across analysis of psychometric data and also using polygenic scores. So the next logical question was whether reduced dietary diversity was associated with microbiome diversity, and in turn, if microbiome diversity was associated with stool consistency. So firstly, asking, is dietary diversity associated with microbiome species diversity? So here we look to see if there was a relationship between Shannon index for food level items, so dietary diversity, and Shannon index based on the species level taxonomic diversity. And as you can see, there is a significant correlation, and it is highly significant. We then repeated this analysis using the Australian Recommended Food Score, and again we see a consistent relationship. So lower dietary diversity, and indeed lower dietary quality, is associated with lower microbiome species diversity. The next question was whether microbiome species diversity is associated with stool consistency. And so stool consistency was measured using the Bristol stool chart, which is a 7-level scale where smaller numbers are associated with firmer, more constipation-like stools, and higher numbers are associated with more watery, diarrhea-like stools. We regrouped this scale to make it more friendly for analysis, and then we analyzed this trait, adjusting for participant group, dietary PCs, sex, and age. And what you can see, I hope, is that having adjusted for these confounding factors, microbiome species diversity, which is indicated here by Shannon, is negatively associated with stool consistency. And the direction of this effect suggests that lower microbiome species diversity is associated with more diarrhea-like stools. And this indeed is consistent with evidence in the literature. So this was interesting because we don't see any direct association between autism diagnosis and stool consistency in the AAB, suggesting to us that this relationship might be downstream of diagnosis. Okay, so second to last slide, and just putting all of this together, what our analyses in the Australian Autism Biobank suggest is that the existing paradigm around the gut microbiome in autism may be upside down, so to speak. So contrary to some reports suggesting that there is a causal effect of the gut microbiome on autism, for example, coming from some animal model studies, our analyses are consistent with a more intuitive, perhaps top-down pathway whereby autism diagnosis and autism-related behaviors and preferences are associated with restricted diet, which in turn is associated with reduced microbiome diversity and looser stool consistency. So in a nutshell, restricted diet drives weak autism microbiome associations. Of course, there are a number of caveats here, and one of them is that although our study is by far the largest of its kind to date, it is still a modest sample. And to our knowledge, there is no equivalent data set that links whole shotgun metagenomic sequencing, genome-wide SNP data, deep psychometric data, and time-matched dietary data that would enable replication of our findings. We, of course, hope that publication of our analyses, which are currently under review, will stimulate the field to generate these types of data sets, and we look forward to the possibility of contributing to larger studies and meta-analyses to explore this really important question in more depth. So I'd just like to finish by noting a couple of implications that we think that our results have for the field, for the autism community, and perhaps for psychiatry more broadly. And the first is that we think that our results should perhaps temper the hype around the idea that the microbiome may be causally related to autism. We think that our data could contribute to forming an evidence base for the community for interpretation of microbiome-based interventions in autism in the absence of double-blinded clinical trials. Our results really highlight the importance of collecting and appropriately adjusting for confounders like diet in analyses of the gut microbiome. And clinically, an important implication is that managing adequate nutrition for children on the spectrum should be a high priority, because a lower microbiome diversity is known to be associated with poorer health outcomes. And then finally, our data is consistent with the idea that autism-related behavioral preferences can influence diet and therefore influence the gut microbiome. And this is likely to be relevant to analyses of microbiome data for other psychiatric disorders like schizophrenia and depression and other conditions. Okay, well, with that, I'd like to finish with some important acknowledgements. And first of all, of course, thank you so much to Chloe Yap, who's done a wonderful job of analysing these data. Thank you to all of our colleagues at the University of Queensland, including Anjali Henders and her team. In the Australian Autism Biobank, there are a very large number of people to thank. I have mentioned Andrew Whitehouse, Velsa Epin, Cheryl Dissanayake, and both Paul Dawson and Helen Huesler, but thanks also to their wonderful teams, without whom we would not be able to do these analyses. We thank our colleagues at Microba and also the Queensland Twin Adolescent Brain Project. And for funding, we acknowledge the Autism CRC, the National Health and Medical Research Council here in Australia, and also the MARTA Foundation. And of course, heartfelt thanks to all of the families who have generously contributed to the Australian Autism Biobank and also the Queensland Twin Adolescent Brain Project, without whom this important study would not have been possible. And finally, thank you everyone for tuning in. It's been a real privilege to present to you today.